Dr. Philip May is actually one of our newest faculty members and probably one of our most accomplished faculty members, all in the same breath. We're very honored that he has moved to Kannapolis to be part of our team, and his research I think you're going to find extremely fascinating. He is very accomplished. Dr. May led the first ever epidemiology study on fetal alcohol syndrome. He followed that first study by saying, you know what, there's a real need, and directed the first major national fetal alcohol syndrome prevention project. That's pretty groundbreaking stuff. And he's conducted his research across the United States, in South Africa, and Italy. I know he's going to tell us a little bit about the studies and his travels, but he's internationally respected as a leader in the field. And that recognition has gone far and wide, and over the summer, he actually was presented with an Excellence Award for his contributions over the course of his career by Senator Tom Daschle in Washington, D.C., by the National Organization on Fetal Alcohol Syndrome. So he comes highly accomplished and highly skilled, and I think you're going to enjoy hearing from him tonight. In addition to his appointment at the UNC NRI, Dr. May serves as a professor in the Gilling School of Global Public Health at Chapel Hill and maintains a role as the extraordinary professor, that's my favorite title yet, he's a faculty member, an extraordinary professor of obstetrics and gynecology, and a member of the Faculty of Health Sciences at the University of Stellenbosch in Cape Town, South Africa. He has co-authored 120 journals, book chapters, and other scientific and public health publications, and we like to claim that he has some deep roots to this area because he began his career just a few miles away from here, and I think he's going to tell us a little bit about that. We're really honored to have him as part of the team. I know you're going to enjoy hearing from him tonight, and it's my pleasure to present to you Dr. Philip May.
Thank you, Beverly. That was nice. I've got both on, so let me just turn this one off. Now, can you hear me now? That's good. As Beverly said, I do have roots here. I'm actually a Virginian, but in 1965 I came to Catawba and graduated in 69. When I left Catawba in 69, I went up the road to Wake Forest for my master's degree, and then went into the public health service, which I'll show, which was the springboard for my entire career. My Ph.D. is from University of Montana. Actually, I had planned to come back and get my Ph.D. at North Carolina State. I had a few other options too, but I had been in the West then for 4 years, and once my wife and I started looking at various places, University of Montana suited us really well. And it just turns out that my major professor at the University of Montana was a gentleman named Gordon Browder, who happened to be also a Virginian, who had gotten his Ph.D. at the University of North Carolina. And it worked out very well. So I do have a lot of educational roots in North Carolina, and I'm just down the road from where I spring from in Virginia. In 1970, after I finished at Wake Forest, I enlisted in the public health service, and that really is what led to this entire 40-some-year career that I'm going to be telling you a little bit about tonight. Basically, public health service opened my eyes to a number of public health problems and issues, but particularly methodology of how to work with some of these problems. And I was stationed in Pine Ridge, South Dakota, and even though it wasn't the best time to be there, Wounded Knee occupation occurred during that time. That was also a good experience as to how to deal with some things that basically disrupt public health in general. So now onto the research. That's enough about me. I'm basically a demographer. Demography is a scientific study of populations that focuses on their size, distribution, age structure, fertility, mortality, death, those sorts of things. And it comes from two Greek words, demos, meaning people, and graphy, meaning the written record of it. But demography is usually, when people think of demographers, they think of the census, which counts people and works with how they move around, but also vital statistics, which are our birth and death records in the United States. A subfield of demography is what I do, and that's epidemiology. Epidemiology is the study of the distribution and determinants of health-related states. Some people think it's just basically the study of epidemics, and that's where it originally came from, but in fact, we study anything that has to do with health and illness. And that's really what I polished an interest in the public health service, and then when I went back to get my doctorate, that's what I spent virtually all my time on. Now, I'm an old-fashioned guy. I'm what's known as a shoe leather epidemiologist. There are very few of us left. Most epidemiology is done in offices via computers now. But shoe leather epidemiology is a colloquial term meaning the gathering of information for epidemiologic studies by direct inquiry among people. It comes from the fact that you have to go door to door or walk around a lot in communities to get to know those communities well, and you wear out your shoes in the process. And that's basically what I do. Now, I probably should be out wearing out my shoes more now than ever before, because I put on weight spending too much time in the computer room. But anyway, shoe leather epidemiology, when I introduce myself at Chapel Hill or wherever, and I say shoe leather epidemiology, a lot of the people my age smile big, but the younger people go, huh? You know, they're not really sure what this is. What I'm going to talk to you about tonight, I'm going to take you on a little adventure. There'll be lots and lots of facts, lots of data here, but I'm going to also show you a lot of pictures about the realities that I study. And we're going to look at first the basics about fetal alcohol spectrum disorders, what they are, how they're diagnosed, and what they look like in terms of patterns in various communities. Then I'm going to talk a little bit about the epidemiologic characteristics of children who have fetal alcohol spectrum disorders, what they're like in terms of their physical features, what they're like in terms of their behavior and their cognitive and behavioral abilities, but also what their mothers are like that have given birth to them. And we'll look at that as those patterns exist in the countries that I study in the most, the Western United States, but also South Africa, where I've probably logged more time studying it there than anywhere else, and also Italy, where we've done studies. Now, it's a very simple concept. That is, the mother and baby essentially have the same blood alcohol levels. So if she drinks a beer, if she drinks a mixed drink, if she has a 4 or 5 ounce glass of wine, she'll produce a particular blood alcohol concentration of, say, 0.02 grams per percent. If she has several, she'll produce one of 0.05, 0.06, getting up towards where it's illegal to drive. The baby will also eventually, with a little delay, produce the same blood alcohol concentrations, but the baby is so much smaller that the actual concentrations are increased by multiples. And moreover, the female has an active liver and can eliminate it about 0.01 grams percent per hour, but the baby does not. And so the alcohol, which penetrates through the placenta into the baby's bloodstream and into the amniotic fluid, the baby is essentially bathed in even higher levels of alcohol than the mother. And I could tell you some stories about that, and I probably will when we get into the South Africa one. But the reason that it's a problem is that alcohol is what we call a teratogen. A teratogen is anything that can cause birth defects. The major teratogen that we usually think about is radiation. If you're pregnant, you will not be given major X-rays because radiation is a teratogen. Well, a number of different chemicals, a number of different drugs, and including alcohol, are teratogenic in that they can cause anomalies or birth defects in a lot of different systems. The face is what is used as a major determinant of diagnosing fetal alcohol syndrome. And most of the features that are different in fetal alcohol syndrome children are external markers of development. For example, probably the most useful is the length of the eye opening, the palpebral fissure measurement. Basically, the palpebral fissure is a measurement of brain development. And if the palpebral fissures are unusually short, then we can have a pretty good idea that the brain has not developed adequately. The nasal bridge is usually very flattened, again, because the growth of the brain, if it's slowed, will not force the nose out and will not force the midface to a point to where it's developing properly. The upper lip is usually very smooth. There's almost no red border to the upper lip. And there's some interesting minor ear anomalies. There are also many other elements of what we call dysmorphology. That is, there are certain bones in the hand, which certain levels of drinking can damage those to the point where with real heavy drinking, sometimes the babies have their fewer flexion points or fewer joints in their little finger. There are abnormal hand creases that develop because of the way the baby moves, because the metacarpals didn't develop right. So there are a lot of different subtle features that for years that have been used to diagnose fetal alcohol syndrome. Now, fetal alcohol syndrome has been around for as long as we've had alcohol. However, the formal diagnosis of it was not put together and made popular until 1973. So there have been millions and millions of babies who have had some degree of compromise over the centuries from alcohol. But what's interesting is that with all of the high-tech equipment for brain imaging and particularly functional imaging, a lot of the features that my dysmorphology colleagues have been using, like the small eye openings, are showing up as major determinants of brain function. All parts of the brain are damaged if the drinking during the pregnancy is constant and heavy. So it's not just the cerebral cortex, it's not just the hippocampus, it's not just one area of the brain. All areas are damaged if, in fact, the drinking is on a regular basis. But what's interesting is brain imaging studies are now showing exactly how that is. And you know what? Those old dysmorphologists who were just diagnosing from palm creases and measuring eyes and things like that, they were right all along, which is really interesting. Because that's how we do it out in the community. I'll show you. Now, Fetal Alcohol Spectrum Disorders is the name that is currently used to describe the fact that there are multiple levels of damage. They can be very mild, and in terms of the dysmorphology or the abnormal physical features that are produced. And those are usually called ARND, alcohol-related neurodevelopmental deficits. The most severe form of fetal alcohol spectrum disorders is fetal alcohol syndrome. And that's where the kids have major dysmorphology. They're very different from the normal size and the normal facial structure of other children. And that's what we first were diagnosing because, in fact, that was the easiest to pick up. These kids do look abnormal, and they do look very different from their unexposed brothers and sisters who have developed more normally. Partial FAS, the children have some minor anomalies, but not as severe as FAS. Alcohol-related birth defects are rarely found, and here's why. Alcohol-related birth defects are where the child has a few of the physical features of fetal alcohol syndrome, but they don't have cognitive, behavioral, and IQ delays. They are in fact, physically deformed, but they do not have the mental deficits, the cognitive deficits the other kids do. Well, here's why. Alcohol is most likely to damage the central nervous system because the central nervous system and the brain are at risk the entire 9 months. So any drinking during the 9 months will cause some damage in the IQ performance, either verbal or nonverbal. It will cause deficits in terms of their behavior. They generally have more behavioral problems. And there are other issues like executive functioning, decision making, memory, attention, those sorts of things that come up. So anyway, most of the spectrum has cognitive and behavioral problems, which I'll show you, but it also has these physical features. Now, for a diagnosis of fetal alcohol syndrome or any of these fetal alcohol spectrum disorders, you have to have evidence in 3 different domains. One, there has to be slow physical growth and development. That is, the children are usually in the lowest 10 to 25th centile of growth when compared with other children at their own age. They also have to have this facial dysmorphology and some of the other dysmorphology, like in the hands, elbows, or other parts of their body for a diagnosis of fetal alcohol syndrome or partial fetal alcohol syndrome. Secondly, they have to have psychological or cognitive and behavioral problems, and that is, they perform less well than other children their own age. And finally, there has to be evidence of maternal drinking. Now, that can be direct evidence or it can be indirect. Usually, we're able to get it directly from the mother's testimony or her interviews. But what our dysmorphologists do, and I have the best clinical team in the world, what we do is my dysmorphologists are also trained as human geneticists, and they know virtually all of the other types of genetic anomalies that produce the same phenotype. In other words, kids who look the same. And they are very careful to rule out other known teratogenic effects or genetic effects that could have caused the child to look, behave, and or be as they are. And so it could be something like Williams syndrome, Down syndrome. It could be Cornelia de Lange syndrome. There are a number of syndromes where the children look the same. But in fact, we are very careful to rule those out before we will even consider a diagnosis of fetal alcohol syndrome. This is an example of the most severely damaged child's brain ever that any of us have ever seen. The brain on the right is the brain of a fetal alcohol syndrome child who died at age 5 days because of the severe damage in the central nervous system. But the one on the left is in fact a normal brain, and you can see all of the convolutions in the brain, all of the sulci and gyri are in nice positioning, and the surface area that they have to think with is great. The gray matter is on top where it's supposed to be. And you find none of the same situation with the fetal alcohol syndrome child's brain. The children with fetal alcohol syndrome and partial fetal alcohol syndrome, and now we're finding with imaging, ARND kids have a number of different places where there's heterotopia, that is scrambling of the 12 different layers of the brain. And there actually is a very decreased cell mass because there's been a lot of apoptosis, which is a type of cell death, which is very common and caused by alcohol. And so they really don't have the proper equipment with which to think. Early in the 70s, right after the diagnosis was formed, most of the kids diagnosed with fetal alcohol spectrum disorders had full-blown FAS, and they were severely compromised. We thought that all kids with fetal alcohol syndrome looked that way. But in fact, we're finding now with the spectrum disorders that indeed there are a lot of different faces that show up with fetal alcohol syndrome, so that you can't always spot the severe ones even as they walk through the room. The partial fetal alcohol syndrome kids and the alcohol-related neurodevelopmental delay kids are easy for my team to diagnose anymore because we have a decided battery which we can use to discriminate them from other children, but in fact, they do look quite different in many ways. Now, one of the things that we found, and we were actually very proud that I found this, and one of my colleagues, we found this, that we took this slide in 1978, I think, and we found that basically the severity of fetal alcohol syndrome will increase in each child born after the next one. So in other words, these children up here are the 6th, 7th, 8th, and 9th child born to this mother. And if you know what you're looking for and you take the measurements, each one is much more severely damaged, each one is more severely impaired. And we were the first ones to publish this, so we thought we discovered it. Well, I thought that for a couple years, and then I was reading an old article from 1899, and there was a physician in England named Sullivan who actually, in very different and very archaic terms, described exactly the same pattern to children of women who were in prison for being drunkards in England. But anyway, during the gin epidemic. So, I think it's very humbling that there really is nothing new in science. We just continue to rediscover what a previous generation or two of scientists had known.
Yeah. What does that mean about the drink? If the women were in prison, were they still drinking?
No.
Okay, so it doesn't take continual alcoholism by the mother.
These were not from prison. The Sullivan was. What Sullivan did was...
The mother continuing to drink through all of these 9 years?
She was. And if they drink it anywhere near the same level, the damage will be equally or greater in its severity with each subsequent child. So in other words, the 4th child born to a mother, if she's drinking at the same level she drank with the 3rd, will produce higher severity.
But if she's in prison and she stops...
Well, Sullivan, the way he did it, Sullivan noted that the women who were imprisoned were having children that were much better looking and healthy than the kids that they were bearing out of the prison. And often they had had several stillbirths out of prison or miscarriages because the alcohol was doing so much damage. But when they were in prison and they weren't able to drink, they were much healthier and they were actually being born instead of miscarrying. And so he went out in the community and studied that. Okay. This is a slide one of my colleagues took at the request of this mother, and this is a very, very severely affected child with fetal alcohol syndrome. And her comment is very common with a lot of women who have severely damaged children. If someone had only told me about FAS, and I wish it were that simple, but in fact it does work with some people. But I'm showing you slides that really cannot be released outside of this room. And these are just some of the children that we've identified and worked with over the years. Now, it was mentioned by Beverly that I was given the opportunity to do the first epidemiology study on fetal alcohol syndrome, and that was in 1979. I applied for and got a contract with the Indian Health Service to do the first epidemiology study. And what we found there is the estimated rates of fetal alcohol syndrome at that time was 2 per 1,000. And what we found in the 3 different tribes here, that is the Navajo, the Pueblo, and the Southwestern Plains, which were several groups of Apaches and Utes, that the rates varied tremendously. And sure enough, when we went back and looked at the drinking pattern, the Apaches and Utes, the Southwestern Plains, had a much higher rate of drinking, particularly binge drinking. And binge drinking, that is, consuming 3, 4, 5, 6 drinks in a day, is the most damaging form of drinking to the unborn baby. Why? Because it produces such high blood alcohol concentrations. Whereas 1 drink a night, 2 drinks a night, there are actually epidemiologists in Europe who say that not only is safe, there are a couple articles that said it's actually produces smarter kids. I don't believe that because I think their studies are flawed because they don't control for maternal education, they don't control for socioeconomic status, and the kids born are being tested with tests that they're going to benefit no matter whether they're damaged or not. They're going to benefit from the home environment with highly educated parents, and they're going to develop well. But anyway, the point is that we found very different rates of fetal alcohol spectrum disorders. FAS and FAE here was about 2% in the highest rate tribe, but in fact only about 3 to 2 per 1,000 in the other ones, which surprised a lot of people. They thought that there were much higher rates. But this is an example from another one of our studies with North American Plains Indians. Why have we studied North American Plains Indians over the years? It's because the most popular drinking pattern, even though only half of the women drink at all, Half of them just don't drink at all. But the ones who do drink, there is a fairly high percentage of those who binge drink. They don't binge drink every day, but they'll binge drink 3, 4, 5 days a month. And but it's real heavy when they do. And anyway, the point is that we can clearly see the damage. We can clearly learn about the spectrum. And this is just an example. This is a dysmorphology score. This is the total number of anomalies that they have on average. Fetal alcohol syndrome kids have 15.4 for a dysmorphology score. The partial are not quite as damaged in terms of the physical features, 11.4. The controls have some minor anomalies. We all have minor anomalies, but of 4. So it's what this all tells you over here, that's statistically significant. The lighter brief IQ and the DKEFs, which are all measures of cognitive proficiency, and you can see that they're all significantly different. The FAS kids and the PFAS kids doing less well than are the controls, which are right around average. Same thing with all these DKEFs. PPT is the progressive planning test, which is a measure of decision-making or executive functioning. And again, the fetal alcohol syndrome children are doing less well. It's significant. The more advanced PPT trials, they're doing even worse. And digit span is another measure of executive functioning. So the point is that they are impaired physically. They're also impaired in terms of their ability to function, in terms of their thinking, decision-making, and in terms of their overall behavior. Now, why do I do fetal alcohol syndrome research? Well, Beverly also mentioned that I was able to start the first major fetal alcohol syndrome prevention program in 1983, the National Indian Fetal Alcohol Syndrome Prevention Program. We trained in 93 different Indian communities across the nation and put out a lot of information. And the first frustration that came from that, we had done our epidemiology studies only with 7 different communities in the Southwest, and we go out to these other communities and they say, well, gee, that's important that we prevent FAS. They were very enthusiastic about that, but they'd always have a couple questions like, well, how much of it do we have here? And if we know how much we have here, then maybe we'll know if our prevention works. And that was so frustrating to me because the contract that I had to fund the thing, the federal government would not let me do any research. It had to be all training and primary prevention. And primary prevention is the least effective. Basically, it's putting out information. And if somebody has an addiction or an alcohol abuse problem, they're usually not able to act on that information. So it was very frustrating, that first program. But the point is, this is an Institute of Medicine model of prevention, which they put out in 1996. And they were very careful to point out the core research where you understand the patterns of the problem first, then you can move to prevent it more effectively. So I've been doing research on fetal alcohol spectrum disorders for a lot of reasons. One, just to find out the basic etiology, but 2, from this basic etiology or causes that we find, we can design programs to prevent it. And I'm not going to go into prevention much tonight, very little actually, but we could do a whole nother session on that sometime. Because we've had prevention programs all over the United States, and we have a huge trial going in South Africa right now. And I'll show you why South Africa. In our epidemiology studies, we have a special way of sampling. We find that in-school studies of first graders are the best way to determine the prevalence of fetal alcohol spectrum in a population. So what we do is we go into first grade classrooms and we measure all the kids for height, weight, and head circumference. We oversample the small kids because most of the FAS kids will be there. And right now we tend to oversample all kids who are in the 25th centile in height, weight, or head circumference or below, because the bulk of the kids who are affected will be in that size range. Then we pick randomly selected controls so we know what the normal is in that community that we can compare them to. And then we measure all of them through a major dysmorphology exam where we produce one of those dysmorphology scores. If, in fact, our dysmorphologists, our pediatricians feel that the child might have some level of fetal alcohol spectrum disorders, they refer them in and they get neuropsychological testing, similar to what I showed you in that last slide. But then we also go out and interview their mothers about what they did in terms of risk factors for the baby during that particular pregnancy. Then once we get all that together, we have a major case conference where we have psychologists, educational diagnosticians, the pediatric dysmorphologists, and our maternal interviewers all together, and we review all of the information. It's 300 or 400 data points on each kid. Then a final diagnosis is made, anywhere from not fetal alcohol spectrum disorders, which is the most common diagnosis that we make, thank goodness, but anything from fetal alcohol syndrome to alcohol-related birth defects. So this is one of our case conferences in Albuquerque. This one's probably 5 or 6 years ago, but sitting around the table are 3 medical geneticists, actually 4, one from Italy, and several psychologists, educational diagnosticians, and maternal interviewers. And that's how the diagnosis is made. So it's a multidisciplinary diagnosis. If you ever take a child in for a diagnosis of any of these birth defects, but especially FASD, you'll want to make sure that they are doing it from a multidisciplinary perspective. Entirely too many of these kids are labeled or diagnosed without a full complement of research of examination from all 3 of the domains that I showed you. This slide just shows you some of the in-school studies we've done. I noticed that we have a big one finishing up in South Dakota right now I didn't put on here. But we have given these dysmorph exams to over 6,000 kids just in this slide, and we have diagnosed almost 1,300 cases of FASD just in these in-school studies. We do other studies too on referral basis, but my clinical team has more experience with this diagnosis, I think, than any group in the world. It's certainly been the most intense experience. We use Institute of Medicine recommended criteria, which were hashed out over a period of 2 years, 1994 to 1996, by a number of experts from a number of disciplines that came together. And we are constantly trying to update the diagnostic criteria. The reason is that there's still a lot of people out there who have their own systems. There's constant debate, fights about what is the best way to diagnose fetal alcohol spectrum disorders, where the cutoff points should be. But we're always working at better diagnostic criteria and better ways, better evidence to make it from. One of the things that we've specialized in over the years is there is this understanding, it's a myth, and myths usually have some truth but a lot of untruth, that you can't get accurate information from women about their drinking during their pregnancy. And there is a lot of truth to that if you're interviewing them in the prenatal clinics when they're pregnant and they think that there's going to be a consequence if they tell you that, well, yeah, I have been drinking a couple drinks a night. There's also, if you're dealing with middle and upper class women who know what fetal alcohol syndrome is and the questions aren't asked in the right way, that you're going to get inaccurate information. But we have specialized in, we've been experimenting with different questionnaires over the years where we sequence the information so that it's more accurate, but it's also more honest. And what we do in our interviews is we review their general health issues first and foremost, because alcohol's not the only thing that affects the health of a baby. There are many, many other things that do. So we start with their general health issues, and then we measure the mother's height, weight, and head circumference, because there are a lot of women who genetically, just their body composition, they are smaller. And we want to use that as a norm to predict how big the next baby should be, or the baby should be, I should say. And then we review daily diet and nutrition. And you're probably wondering, up to this point, why the heck am I at a nutrition research center? Well, we're getting into that. We review the daily diet and nutrition. And we started doing those interviews And I'll be frank with you, as a smokescreen, I know nothing about nutrition. At least I didn't then. I'm learning more all the time, especially now. But we just use it as a smokescreen. So I have a lot of data that we have not analyzed about the nutrition of women in South Africa and Italy and the United States. And but what we found was that the women who had the most severe fetal alcohol syndrome and fetal alcohol spectrum disorder babies were what? They had a very low body mass. They were skinny. And in fact, there are a lot of things that point to the nutrition link, and nobody has really gotten onto it much, even in animal studies. Why did we catch onto it? Well, in 1999, we had just finished our second in-school study in this one town in South Africa, and our geneticists decided, well, let's look at all the kids in a family. So we brought in whole families of 5 and 6 kids, and mom and dad, and sometimes grandma would come in. And it was really interesting. We also brought in some other families who didn't have fetal alcohol syndrome kids. And the mothers were like night and day. The mothers who were big and robust of the normal kids, but the small ones were the mothers of the FAS kids. So this nutrition link keeps hitting us in the face from all different directions. And so anyway, in that daily nutrition scan, we would slide into the whole topic of what they drank, from water to lemonade to whatever, to tea, but also alcoholic beverages, and we do that through a daily log. Well, gee, you had green beans and some chicken yesterday for supper. What did you have to drink with that meal? Well, I had some water, and we had some wine too. You know, that kind of thing. So anyway, the sequencing is really very important to get accurate information, and especially in middle class and upper class population where there is almost an incentive not to tell the truth, particularly in the presence of a healthcare provider. So then we go into current drinking, and we do what's called timeline follow back, and we get a number of different things. There are 4 variables up here. The quantity, frequency, variability, and timing of the drinking are very important. We want to know how much they drank, how often they drank it, how regular that pattern was, and when the timing was. Did they drink a lot in the 7th to 12th weeks of pregnancy? That's the most important time as far as the baby's physical features are concerned. But actually the central nervous system, like I say, the brain and the spinal cord are at risk the entire 9 months. So we want to get a handle on that. So that's what we do. And we do very, very detailed maternal drinking questionnaires, whereas a lot of the researchers just assume, well, we can't get accurate information. We're not even going to ask that. We're just going to diagnose from the physical features. And that's probably not the best thing to do. But anyway, so we can get information from the medical chart. We can get information if the mother's brother has died in an alcohol-related motor vehicle accident, if their sister died from cirrhosis, if any of those things. Those are also indirect measurements, but we can get the direct measurements 95% of the time. Let me just take a minute, and I want to show off my multidisciplinary team. There are 5 slides here. All of these people have worked with me. These are just a short list of the dysmorphologists who worked with us over the years. I have statistical and data people on my team, including, for those of you who are psychologists, Barbara Tabachnik has worked with us for years. She's written all of the major textbooks on probability statistics that are used in a lot of different disciplines. Barbara's 74 years old now, and she's had to give up belly dancing. But she's still a great, honestly, she was a belly dancer for a while. She's a little eccentric. But anyway, she's a great statistician. I've had lots of good principal investigators in Italy and various parts of the United States and South Africa. I've employed lots and lots of developmental specialists, psychologists in a number of continents. My field personnel can't be beat, especially in South Africa. I have the most exceptional people who talk about shoe leather epidemiology. These women in South Africa, for example, who have social work and psychology and nursing backgrounds, they're not afraid to go anywhere. And I didn't put it in here, but I have a picture of one South African farm where the sign in the entrance to the thing, into the farm, says, "Warning, trespassers will be shot on sight." Doesn't bother my folks. They go in and get the information. It's amazing. And finally, all kinds of field personnel again that have worked with me, and so all of the richness of what I'm presenting comes from this mass of really dedicated, talented professionals that just are just the cream of the crop. Now, why South Africa? Well, here's why. I've been working in South Africa since 1996. And I was taken over there by the National Institutes of Health as a team of 13 people to lecture and to see if, in fact, maybe fetal alcohol syndrome was a problem. Well, why did we think it was a problem? Well, there was this guy named Dennis Viljoen, who was a dysmorphologist, human geneticist, who came over to the United States and said, you know, I think 35% of the kids I see in my special clinics have fetal alcohol syndrome. And I think I'm crazy. I want somebody to come and help us verify this. So we went over there, and sure enough, there were kids all over. And here's why. The population in South Africa that we've been working with is, one, purely alcohol exposed. There really isn't, at least at that time, there was not a big problem with any other drugs. Secondly, binge drinking is so regular in the towns where we work. People don't drink but 2 to 3 days a week. Friday night, Saturday afternoon and night, and if there's some left over on Sundays. So it's a very regular pattern. Produces huge blood alcohol concentrations in these kids. And that's the major form of recreation. There just isn't much to do other than that. And so the rates of fetal alcohol syndrome are extremely high. I will show you. But remember, I talked about how the baby will maintain a higher blood alcohol concentration longer than the mother. Well, one of our maternal interviewers was interviewing a lady just a couple years ago in Afrikaans, the language over there. And the woman said, she explained to the woman after the interview, here's what fetal alcohol syndrome is. And the woman looked at her and said, very innocently, she said, oh, that's why my baby never moved till Thursday. And then she started drinking again Friday. So the kid was affected for 5 days at a time, and so it's a pretty amazing place. It's you hate to use the term laboratory, but it really is an amazing human laboratory. And just so nobody thinks that we're exploiting these people, that's why we have the biggest prevention program in the world, because as we do the research, we have an obligation to devote a substantial amount of our resources to alleviating the problem. And one of our colleagues, Dennis Viljoen, to be exact, just reported in the literature a 30% reduction in another town in the Northern Cape province, which we work in the Western Cape province of South Africa.
Are you buying this alcohol in the store?
Yes.
Is it the same type of alcohol we would have here? Is the factories or the breweries that make it there?
That's a good question. You make a good shoe leather epidemiologist. Let me show you. I'll skip ahead to that. Here are some of the kids that we saw in '96 when we got there. We picked these 7 kids out of 2 classrooms in a preschool, and the guy on the right there, whose face you see, is Ken Jones, the originator of the Diagnosing of FAS. And I can't quote him exactly what he was saying to me as I took the picture, but he said, "Do you believe this blank?" He couldn't believe how many affected kids we were seeing. There's Ken Jones with our team in one of our early clinics. I'm going to get to the answer to your question. So bear with me. In our epidemiology studies, we do first grade children. Why first grade children? Because they're 6 and 7 years old. And at 6 and 7, there are discriminating enough tests that we can use to distinguish between kids who are affected and those who are normal. The tests are sophisticated enough, they can handle it, that we can discriminate it. And my joy in all of this is in this last bullet. Mine is getting to know a community, which you're asking about right now, getting to know a community, understanding it, and analyzing it in a way that the proof is indeed scientific, the proof is indeed valid, to answer valid public health questions as you just asked. So I won't go into this in great detail, but these are child variables. And what you can see here, just like in those Plains Indian data that I showed you, the The children with fetal alcohol syndrome are shorter than the normals. Their weight is lower than the average kids in that community. Their head size is significantly lower, 48 centimeters as opposed to 51. Their eye openings, again, a measure of brain development, is significantly smaller. They have very smooth upper lip, and they have very narrow vermilion, the red border to the lip, which is also an indicator of brain growth. Their total dysmorphology score is much worse for the FAS kids. And if you look at it on a bar chart, you can see for their their cognitive behavioral performance, the FAS kids perform much lower than do the, significantly lower than do the non-FAS kids on verbal IQ, nonverbal IQ, but their behavior is worse. That's why the bar is higher there. And I can show you this study after study, this pattern repeats itself. The kids are doing much worse. So let me skip ahead to answer your question. So when we went in and we go into a community and we screen the kids, then we have to get out in the community and interview their moms. Here's what the Western Cape looks like. Here's the kind of jobs that they have. They work in the wine producing industry. They work in orchards. They work in other vegetable production. There are some factories around like tanneries. There are dairies and things like that. And the women perform a lot of the labor. Drankvinkel is the Afrikaans word for bottle store or liquor store. And my job was to quantify what they're drinking. So you were asking. It's mainly commercial. But when I got there, we were all told, well, what causes this is the wine. The wine that these awful farmers give them every night or every weekend to drink. And like most industries, that was part of their pay. It was called the system of the DOP system, and it's gone on for over 300 years, ever since the Dutch and the Huguenots got there and started the vineyards. For years, up until the mid-50s, there was this DOP system where they would give them alcohol as part of their pay. They'd also give them, other products of the farm, whether it was vegetables or whatever. But we were told that was what was still causing it. What we found out wasn't. That pattern of nighttime and weekend drinking has persisted, but now it's all commercial. So I have to go out and find out what the alcohol content of these various beverages are. A pop sack is the foil innards of these wine boxes that we have in the United States, but they don't have the box over there. Anyway, this is 5 liters for 15.5 rand. At the time I took this picture, 15.5 rand was about $1.25. 5 liters of wine. So alcohol is very cheap. But the beverage of choice in South Africa and everywhere we've worked that produces fetal alcohol syndrome kids is beer. More kids are produced with FAS than beer. It's not on beer rather than anything else. It's not because it's any worse for the kids. Any kind of alcohol is teratogenic. But in fact, beer is the beverage of choice for the kind of individuals who do recreational binge drinking. And so what we do is we quantify it like this. We take the commercial ones, and then the ones on the right there, those are homebrew kind of things that are made by a lot of the African immigrants into South Africa. It's sold, and we actually would get samples of that and measure the alcohol content so that we know what we're dealing with. And our interviewers would go out with these kinds of a key to be able to calibrate what the women were drinking. Same thing with cigarettes. Cigarettes in this part of South Africa are hand-rolled, and a high percentage of the women smoke, but they don't smoke that many a week. The average in most of these studies is around 38 to 41 cigarettes a week. That's, I remember North Carolina 40 years ago. I mean, most people did that in a day, 2 packs a day. Anyway, so that's what I do. I go out and calibrate these kinds of things, and we set up the interview protocols and do all that kind of stuff. in addition to designing the samples and that kind of stuff. But anyway, one of the things that we found that basically mothers of fetal alcohol syndrome kids are consistently older than the normal controls when we pick the controls randomly. 29 as opposed to 25 and a half in this particular sample. In the United States, it's usually more like 29 to 32 is the average age of a mother who's produced an FAS child, and the average is usually around 25 for the non-FAS. They have had more pregnancies. That's what gravidity means, is pregnancy. 3.6 pregnancies as opposed to the normal controls have had 2.9. They've had more live births, 3.2 to 2.7. Miscarriages, they've had more because alcohol does cause miscarriage and stillbirth. They've had more stillbirths, and all these are significant on a statistical basis. And the child with an FAS that we're studying usually is much higher in the birth order, 3 or greater usually. So anyway, those are the kinds of things that we had to find out in each individual community. Now, drinking during the index pregnancy, the second line item here, 96% of the mothers of fetal alcohol spectrum disorder kids admitted to us and told us details about their drinking. And even 24% of the control mothers were drinking because they were randomly selected. But the point is that you can do some drinking, particularly the mothers, and I'll show you that in a minute. The mothers who drink less, and if they're big and robust and healthy and well-nourished, they can get away with some drinking, and we can't diagnose their kids as having any compromise. Smoking, again, 41 cigarettes per week. And that was fun. I bought those little packages of tobacco that you saw there, not Bull Durham, but that's the packages that they use. And in South Africa, the population that we work in, they use newspaper as their papers to roll it. So I bought those packages and gave it to 2 or 3 of the women in the community where we were staying, and I said, can you roll this just as if you were going to smoke them all? So we'd roll them, and then we'd measure the actual amount of tobacco in each one, and sure enough, it came out, averaged out to a gram. And so, I mean, that's how we calibrate these things, so that we're not just, no pun intended, blowing smoke and giving out bad information. Anyway, but the same kind of pattern with the verbal and nonverbal IQ exists in the South African studies, and we can correlate it with drinks per week, drinks per drinking day, 3 or more drinks per occasion, etc. You can see that the dysmorphology score, or the physical anomalies that the kids have, correlate very highly with the quantity drinking. 0.7 is about a good correlation as you get in these behavioral studies. And basically, the quantity that the mother's drinking correlates very strongly with the anomalies. But also, you can see that the behavior is compromised as well, not as highly. So I won't go into that. Preview to the final part, the prevalence in South Africa is the highest in the world, and we don't take any pride in that, but we do take pride in the fact that we've uncovered it in a way that there are all kinds of very good things happening with awareness and prevention. But the rate of FASD, this is one of our most recent studies, 2008 to 2010. The rate of FAS among all the kids enrolled is 13.6% in the schools that we study. If we just look at the sample prevalence, the smaller kids, the oversampled smallers, it's as high as 21%. So if somebody were to ask you the prevalence in this particular town, it's between 13.6 and 21%. That's absolutely unheard of, which I'll show you later. Here's another study that did 2009, 2011, which we're writing up now. This is an even more impoverished area. It's more rural. It's 3 hours from Cape Town. Similar farming kind of community in small towns. 18 to 25%. Unbelievable. Now, I won't go into this, but this is what I have to do to convince other researchers that, again, that we're not making a bunch of generalizations without the data to back it up. Most everything that we present in the studies like I just showed you, the case control studies, people can say, oh, well, those are just associations, or those are just differences between groups. How do you know that one variable is more important than the other. Well, you do these fancy multiple correlation studies. This is called a structural equation model. And if we just look at drinking, how it predicts the cognitive and behavioral traits, that is the nonverbal IQ, the verbal IQ, and the behavior of the child. If you look at drinking, how binge drinking and quantities of drinks per day, etc., how it predicts the child's behavior, this tells you that drinking alone only explains 17% of the child's behavior. What else explains it? Well, how educated the mother is, whether the parents play with the kids, whether the kids get adequate nourishment, whether the environment is stimulating or intellectual. All of those things explain probably 83% of the variance. So if I presented this data alone to other scholars, they say, well, see, you're only explaining about a sixth of what's going on here. So we include other variables in these equations, and you have to have large numbers, and this is, again, the beauty of, unfortunately, of South Africa, is that we have lots of kids that are affected. So if we include the mother's physical status, if we include her socioeconomic status, like her education, whether she lives in a rural area or not, we include her childbearing status, that is how old she was when she bore the child, how many pregnancies she's had, and those sorts of things. And then we include the drinking as one of the four major variables. We see how those all intercorrelate, and then how they explain the neurobehavioral traits of the kid. By including those other variables, we're up to 55% of the variance. In public health, when you get over 40% of the variance, it's a slam dunk. It's really good stuff. 55 isn't bad. We're real proud of that. And the point is that it's multiple factorial, but yet we still have a lot of researchers who say, well, it's all alcohol. No, there are many other things. And the beauty of that is if there are other things, we can manipulate those other things in isolation from the alcohol and maybe prevent the damage. Okay? So anyway, here's where we're explaining the child's dysmorphology, that is their abnormal features. And there's a much more direct influence here. We can explain 62% of the variance by these same maternal variables. So the child's physical abnormalities, 62% of it can be explained by this. And what's so unique about these models is that we have extremely good maternal data because we're not afraid to ask the questions and we've sequenced them in a way, then we can explain the physical things. It's very unusual to have both of those data sets to be able to put together. So that's why my team is so amazing, because they work together to collect all this information and puts us with really the best information going. Now, we currently in South Africa, we have 4 additional waves of in-school screening going on. My money in South Africa, it all comes from the National Institutes of Health. And we can get studies done on about 1/4 of the amount of money in South Africa as if we can here, because their salaries are cheaper. Just basically the economy is such that the American dollar goes a long way. And there's no better place to learn about this. But we have a proposal in to get another 5 years funded in South Africa at actually a lower level than what we're currently funded. And we're just praying that we're going to get funded again. But here's an example of some of the prevention that's been kicked off by the local community in response to our field workers' attention. This is Fetal Alcohol Syndrome Day. This was the 4th annual in this community with the highest rate in the world. And Fetal Alcohol Syndrome Day, what the banner says up there in Afrikaans, it's 100% preventable and 100% irreversible. Well, that's half right. It is 100% preventable if you can get the people to change their lifestyle and quit drinking. That's true. However, it's not 100% irreversible, and that's the beauty. We're finding that early intervention and stimulation and increased nutrition and those sorts of things can help the brain heal itself, not to a full degree, but these kids can develop much better than we thought they could 30 years ago. And so the 100% irreversible in Afrikaans there is only a half-truth. Even the police are getting involved in it, the kids in their school uniforms. It's a pretty good deal. We also have longitudinal studies going on where we're trying to follow these kids We have kids diagnosed now from age 6 to 23. We also have a newborn cohort where we're trying to get so sophisticated that we can diagnose these kids as early as 12 and 18 months, even with the more subtle degrees of the syndrome. And if we do that, we can do real early intervention. And what sort of tripped this off was a few years ago, we were screening in this one school where this first kid that we ever diagnosed in South Africa with fetal alcohol syndrome was on the wood cutting crew just down the road. The principal said, "Hey, you remember Popcorn?" That was our nickname for the kid because he was so hyperactive. Popcorn's just down the road. You want to see him? Sure. And so we went down the road, and here was Popcorn. Popcorn looks good, and he's doing well. And he's had major trauma in his life, but he's been well cared for. His mother had died when he was like 2 or 3 years old in a house fire. Most people believe that her, quote, husband or her partner burned her up on purpose. He was raised in by her grandmother till she died. He's been raised by the aunt since, but they're loving people, and they've taken good care of him, and he's doing pretty well. So we kicked off a whole longitudinal study a few years ago to find all these kids where they are now. Again, another picture of our South Africa team, looking stunning in their clinic shirts. And here's where I just want to make this point. Early diagnosis does lead to better chances. And the woman in this picture is an educational diagnostician who's worked with me for 17 years. She's trained me well because she's always maintained that if we give these kids an optimal environment, that they will develop nicely. And this shows it. Basically, what this is a language and literacy test where you teach the kids new language skills. And for years it was thought, well, they can't retain this information. But what we show here is the high bar here is fetal alcohol syndrome kids, full-blown fetal alcohol syndrome kids, 20 of them, who got this language and literacy training, and they made dramatic gains of almost 25% improvement in just a 9-month period. The other fetal alcohol syndrome kids who got a cognitive behavioral approach made a nice 10% gain, but it was nothing like this one. And then the non-exposed, the control kids, also made the 10% gain, but you can see that the FASD kids did best. So we've done the same thing in Italy, and let me just, where's the school in this picture? In Italy, we did a random sample of 25 schools over 2 different years. So when we chose the sample, basically it was 43 different schools. Finding your way around the small cities around Rome was just unbelievable. The school is right here. But, I mean, they were just impossible to find. But anyway, in Italy we found very much the same thing. The point was, though, that the traits that the kids had were more on the mild side. The mothers were drinking, but guess what? They were more adequately nourished. They had a bigger body mass. And so the kids weren't as damaged. They also had a lower birth rate. The average number of children that a woman has in Italy is 2, whereas in South Africa it was 4.5. So you don't get into the highest risk later pregnancies. But still, they were drinking wine, and because of some of the socioeconomic and nutritional advantages, the kids weren't as damaged. So the dysmorphology scores were substantially lower. Remember, the South African kids were like 18 dysmorphology. Here they're 12.5. And the controls are even lower. So even though all the kids in the Italian schools were hyperactive, boy, I mean, if you think American kids are hyperactive, go into an Italian elementary school. The teachers and the kids are all just really moving fast. And anyway, and the cognitive behavioral deficits were relatively just as bad, but again, not as much severe scores. And we used Italian tests there, which were compatible to the United States. And let's see. So that's enough for Italy. Now, why did I come to NRI? Because I've already admitted that I'm quite nutritionally ignorant. Well, I've explained that we keep seeing these signs that the mothers who are better nourished, who have a larger body mass, tend to have children who are less severely damaged. We are seeing as well although we have not done any trials other than the one that I showed you on language and literacy. We think that if kids are given both cognitive stimulation and a very adequate diet, particularly of elements like choline and other things, other elements like the omega-3s that feed the central nervous system development, that they can do well and they can catch up. Even though they've suffered from cell death prenatally, the brain still is plastic for a number of years after birth, and we think that they can progress. Now, the simple view of causation is that the child's traits will be influenced totally by alcohol. The quantity that they drink, the timing that they drink it in the pregnancy, and the frequency, how often they drink it. But we have shown in our research, and I've shown you some of the tidbits of what we found, that nutrition of the mother, lifelong and during pregnancy, is extremely important. We have a blood analysis trial as well as 24-hour recall going right now in South Africa. I just got another case in today. Vitamin A is coming up a lot. Vitamin A deficiency. I don't know. But anyway, we have nutrition trials going, and we think that we can influence both the child and the mother through enhanced nutrition. And that's a lot easier to influence than some of these other things, socioeconomic status. I mean, the population in South Africa is impoverished, but it's going to take years to change. By the way, something that we found very regularly in every population we've worked, that the women who have a higher degree of religiosity and spirituality, if they attend church regularly or even pray regularly, have lower rates of drinking. The kids aren't as severely damaged. Just so you know, it's hard. You know, in academics circles, you can't present data like that much anymore. But it's an extremely important part of these individuals' lives that is protective. We kept showing very big differences between the mothers of normal kids and the FAS kids on spirituality. So there is normal individual variation, and here's why I'm here. That is, even with the exact same level of alcohol exposure, FASD trait expression varies greatly with each individual human. And lo and behold, with sophisticated imaging technology now, animal studies like mice or rats that are identical genetically with the same level of alcohol will have different levels of damage. Kathy Sulik, who's a professor of pathology at Chapel Hill, has been a colleague of mine for years. And when I was interviewing for this job a year and a half ago, I had to interview first at Chapel Hill, and Kathy took me around and she took me into her lab and she showed me 3 different mice images of their brains, and one was, by the color, was severely damaged, one was sort of intermediate damage, and the other one was not damaged hardly at all. And I bit. She said, "What's going on here?" I said, "Well, obviously you gave them different levels of alcohol." She said, "No." And I said, "Well, then they're different genetically." "No. Just normal variation." And you get even more variation in humans where we all have different genetic traits. We're not laboratory animals that are bred to be genetically the same. And so anyway, individual variation is why I came to NRI, because that's the focus of NRI, particularly as it relates to nutrition genetics and metabolomics and all of the things that can explain some of this variation. So one of the 24-hour dietary recalls that we were doing as a smokescreen, we've analyzed the data, and we found that both mothers of FASD children and the normal children were deficient in most micronutrients below the daily reference intake levels, except for B12, selenium, sodium, and total omega-3 fatty acids. For some reason, they were okay in this particular sample. But for all South African mothers, many vitamin intakes were less than 50% DRI. Now, what we did do was compare the mothers of FASD kids with controls, and we found these 3 deficiencies in this one sample. Riboflavin was low in the mothers of FASD children, calcium was low, and DPA, one of the omega-3 fatty acids, were low. We weren't able to test for choline in this particular sample. We're looking at choline in a second sample from here, but we haven't found a difference there either. And some of you may know that choline has been shown by our director, Dr. Zeisel, to be very important for brain development. And in the blood study we have going now, we are doing special choline analyses on the blood samples. So what we did is we added that to our prevention protocol. In the United States, we've been doing a dual approach. We've been offering the women who had a fetal alcohol syndrome child, we've been offering them alcohol reduction therapy, brief motivational interviewing, and we offered them, if they were not pregnant, birth controls to prevent a further alcohol-exposed pregnancy, because it's often easier not to have the kids than to quit drinking for women or anybody who's dependent on alcohol. And I directed the largest center on alcohol research in the Southwest for 10 years. I know that literature as well. Anyway, finally, what we did in South Africa is we added a nutritional component in our case management. So women who have a particularly low bicep measurement we figure are nutritionally deficient, and we give them a can of Ensure every day that they're pregnant. We're going to have about 40 cases to analyze by the end of this year. I don't know what the results are going to be, but I think they're going to be positive. We're just using the dual approach in the United States. Out of 120 women that we had in the Northern Plains, only 2 of them, just using the alcohol cessation and approach, only 2 of them, of the 120, had a damaged child in the pregnancy where they were in case management. So we eliminated the majority of it. So I'm hopeful that in South Africa, even though we have a lower quit rate among the women, I think that we might be protecting the kids with the nutrition. So one of the biggest questions has been, is there a threshold for blood alcohol concentration to produce an FAS or an FASD child? Is 1 drink a day safe? Is the threshold, if you have 1.5, then the kid's going to be damaged? Is it 2 drinks a day? Well, in England, they say clearly 2 drinks a day is okay. Beyond that, don't worry about it. Then it's a problem. But anyway, so is there a threshold? And that's been a big question. And here's my, I think, most impactful slide. We can get information on each trimester of a pregnancy in South Africa, and it's very accurate. The women do not distort their reporting much, we can tell. This is first trimester drinking, and the DDD here is drinks per drinking day. In other words, how much they drink on a normal day when they do drink. And remember, these women just drink 2 days a week, sometimes 3, but usually 2 drinks a day. I mean, 2 days a week. The mothers who produced FAS kids were drinking almost 6 drinks a day when they drank. It's Friday and Saturday usually. The mothers of partial FAS kids were drinking 4 drinks a day. The mothers, 24% of the mothers of normal kids, 25 actually, drank, and they drank almost as much as the ones who produced PFAS kids. And you can see that persisted all through the trimesters for the mothers of FAS kids, so it's the repeated assault all during the trimesters. The reason these kids are probably less damaged is not only that they were drinking less, they tend to taper off in the latter parts of the pregnancy, and the central nervous system can still play some catch-up at that time. And the normal mothers, the mothers who produce normal kids, were drinking the same, but their kids were normal. Then the second line here is blood alcohol concentrations. We can take the alcohol information and we can use an algorithm called the Bacchus, which takes into consideration the mother's weight, the quantity that she drinks, and the time that she takes to drink it. Like, does she have 5 drinks in 6 hours? Or does she have 5 drinks in 3 hours, or whatever? Anyway, this algorithm then produces an average peak blood alcohol concentration. And you can see that the concentration is much lower in these mothers who had normal kids, even in the 3rd trimester. Notice that they're about the same, almost 0.2 all the way through. But here, it's 0.12, and it goes down to 0.8, which is lower than these mothers. So the blood alcohol concentration is being dealt with, and this is just a graphic presentation of that. But the alcohol is somehow, these other cofactors are protecting the kids of the normals. Well, here are 4 variables that we know are either risk or protective. One protective factor I've already told you about is body mass index. The mothers of the FAS kids have a body mass index which is classified as lean, 22.5, whereas the normal mothers have a body mass index which is just high normal. It's just getting into the overweight range. Education. The mothers of the FAS kids tend to be less educated. Do they not know about FAS? I don't know. But educated people often can exercise better self-control, and they probably more likely have the facts. Risk factors are gravidity, that is the number of pregnancies. They're in their 4th pregnancy when they have an FAS child, whereas the normal mothers are in their just finishing up a 3rd pregnancy on average. So they're less at risk there. And the mothers are older of FAS kids, younger of the normal controls. So these are 4 differences that explain some of the variance. Nutrition, hopefully I'll have really good data in the next time I give this talk here. In a year or so, we'll have nutrition evidence, whether it's good or bad. One final thing, and then I'll talk about just a few minutes about the prevalence. The genetic control of not only the metabolism, but the genetic control of everything from the bacteria in your body to how you metabolize food, how you metabolize alcohol. All of that has genetic links to it. And there are often changes in the genetics through a process called epigenetics. Well, one way to find out about the genetic influence is to look at twins, particularly monozygotic or identical twins. They have exactly the same genes. But again, remember the individual variation. When we look at 9 pairs of identical twins, monozygotic twins, which we have seen over the years, 75% of the cases, they have exactly the same level of fetal alcohol syndrome. In other words, both girls, both boys in the monozygotic twins have FAS, or ARND. Exactly the same level of damage. So there's something genetic going on that makes them similar. If you look at fraternal twins, dizygotic twins, the correspondence is only about half, 37.5. So there is an opportunity there through genetic variants for alcohol to do much greater variance in its damage. So the implications here are genetic. The other thing, I have biophysiologic as an interesting thing that we need to study. There has often been a belief that children who, twins, the one that is lower in the mother's womb gets more alcohol than the one up top because the specific gravity of alcohol makes it gravitate to the lower parts. So that's another thing that would be really interesting to find out with the new imaging technology with the Doppler. This could be monitored, and blood flow could be monitored, those sorts of things. So there is a biophysiological issue. But then there are also other things. Remember how the women who are older had more severely damaged kids? What's going on? That's a process in public health that they just sort of grossly call weathering, aging. Is it a change in the electrolyte balance in the intestines? Is it the ability to metabolize certain things? Is it change like we saw last week in the gut microbes? I don't know. But anyway, all of those things await. They're really interesting. That's why I came to NRI, in addition to the fact that I wanted to come back to where I came from and back to where one of our daughters has been teaching school in Concord for a number of years. And my son-in-law is Concord native, so that might be heresy in Kannapolis, but yeah, anyway. But we do live in Salisbury. Okay, FASD prevalence. Now, that was the title of the talk, wasn't it? Took us a while to get here, but here we are. And I'm going to skip to just a couple slides here. I've got many more than we need to go into. There have been commonly accepted estimates of the prevalence of FASD in the Western world, meaning the United States and Europe. And those estimates that have been accepted for years, and I actually published some of these just as recently as 11 years ago, were 0.5, in other words, a half a kid for every 1,000 to 3 per 1,000. That was a commonly accepted estimate for full-blown FAS. For FASD, that is FAS, partial FAS, ARND, and ARBD, the estimates have been accepted from one study that was done in Seattle back in the early 70s. And everybody said for years, 1% of all kids in the United States suffer from one of these diagnoses of FASD. Okay? 1%. Well, I think you've seen that in the special populations in which I've worked, it's a lot higher. Okay? But everybody will come up to me and believe me, they've said this for years. Well, that's just those populations. You just run to the sound of the guns. You just go where it's high. That's why it's higher. We don't have that problem in Kannapolis, or we don't have that problem in Albuquerque. Or I can tell you, I've really heard it in spades. We don't have that problem in Sweden. We had a whole study designed in Sweden with our Swedish colleagues, and a couple of their colleagues just slam dunked us. They just, no, we don't have that problem in Sweden. You guys are just diagnosing genetic birth defects. Those aren't really FAS kids. Anyway, basically, do you know the story of the drunk and the street lamp? Well, it's late at night. It's 12 o'clock. Bars are letting out in this particular town, and you're driving home, and you see this guy under the street lamp. And he's down on his hands and knees, and he's groping around. And you stop your car, and you go over and say, what are you looking for? Can I help you? And the guy's obviously intoxicated. And he says, no, I lost my keys. They're somewhere around here. And the guy said, well, did you lose them right here? Whereabouts right here did you lose them? Oh, I didn't lose them here. I lost them over there. Well, why are you looking for them here? Well, the light's better here. It's dark over there. I'll never find them over there. That's what we tend to do in science. That is, we use the methods that we have that seem to work the best under the best conditions. We don't go out and find the cases. So there are 3 different methods for finding cases of most any birth defect, but fetal alcohol syndrome is an example. There are surveillance and record collection systems, most of which come out of Atlanta from the Centers for Disease Control. And it works very nicely for things like Down syndrome because there's one genetic marker, trisomy 21. And you can do a quick blood test. Everybody knows how to do it. Bam, you got a case. So you can recognize those kids at birth. Fetal alcohol syndrome, you can't really tell the difference in these kids, particularly the subtle varieties, until they're 6, 7, 8 years old. So they don't work very well. Clinic-based studies, you interview women in the prenatal clinics, find out how much they're drinking, how much they'll tell you they're drinking, and then you examine the kids when they're born and maybe up to 18, 24 months. There's those clinic-based studies. And then there are active case ascertainment studies where you go out and you find the cases. And we first started going out to find them through special referral clinics, working with doctors, nurses, schools, and say, refer the kids into us. But anyway, basically, this is the rates of FAS produced by the method. Surveillance produces less than 1 per 1,000. On average, median, the midpoint, is 0.3 per 1,000. Almost no cases. Clinic-based studies produce about an average of 2 per 1,000. And you can see on these charts how much they're producing. But then active methods produce 16 per 1,000 or 8.5 per 1,000, which you see here, the median and the mean. So you're producing a lot more. And again, they'll say, well, the active studies are only done in these high-risk populations, like I work in. But then if we look at FASD, surveillance studies produce no FASD kids. Never have, never will. And this is, by the way, about the summary of over 70 studies here. Clinic-based studies produce much higher rates of FASD, of some damage, of 6 per 1,000, still less than 1%, or 5 per 1,000 in the median, so less than 1%. Active methods, and this doesn't include the in-school studies, produce 38 per 1,000, almost 4%, and the median is 2%, so 2 to 4%. So again, drunk in the streetlight, well, gosh, surveillance is the easiest. It's under the streetlight, so we'll look for the keys there. but in fact we should be over there where the keys were actually lost, searching out these cases through active case ascertainment. Well, obviously in-school studies, we're great believers in that. We have done 9 in-school studies ourselves, 9, 10, anyway, and now we've had a couple of colleagues, one in Croatia and one in, a couple in South Africa, one in Croatia and one in Australia that are doing in-school studies now, and they're finding very similar things. I must do a tip of the hat. All of our in-school studies have been funded by NIH, the National Institutes of Alcohol Abuse and Alcoholism, and actually 2 of these people are now in North Carolina. Faye Calhoun actually has a project over here in our building. She's with North Carolina Central these days, and T.K. Lee is heading up a big Asian medical school effort for Duke University. Anyway, they've been very supportive over the years of our ideas. So in-school prevalence studies are the right time to do it, and you find a lot of kids. And in our studies, as I showed you, we look at all areas, the growth, the cognitive and behavioral tests, maternal interviews, and we've done a lot of kids. South Africa, we find these very high rates of FASD of up to 9% is what you see here, and then I showed you even higher ones from our more recent studies because of some fine-tuning we've done in our diagnostic system. In Italy, we found, these are low averages, but 3%, 3.5% we found of FASD in Italy. We've done a pilot study, 3 pilot studies in a city, a moderate-sized city in the West, and we found 1.5% to 2%. And one study we only found 1%, but again, 1 to 2% for FASD. So let's put all this together. It varies by country, and basically we've come up with these new estimates. Basically, we feel that fetal alcohol syndrome is 2 to 7 per 1,000, approaching 1%. And remember, the other rate was 3 that's commonly accepted. We feel that fetal alcohol spectrum disorders affect 20 to 50 per 1,000, or 2 to 5%. So when you look at that graphically, here are the commonly accepted estimates. The high estimate of 3 per 1,000 is just about the midpoint of what we really think it is, more like 7 per 1,000. FASD, instead of being 9.1 per 1,000 or 1%, we feel it's 2 to 5% in the United States and Western Europe. And actually, one of our Italy studies, we actually found 9%, but I'm reporting averages here. So the point is that we feel it's too low. For years, there were some people who ran around and said, fetal alcohol spectrum disorders are like an iceberg. You only see the part that's sticking out of the water, that is the very severe cases. And underwater, there are hundreds and thousands of cases. So only a few FAS kids get diagnosed, a few partial FAS kids get diagnosed, and there are all these kids down here, FAS and partial FAS, that never get diagnosed. And I could tell you some stories about that too. But anyway, conclusions. If we don't actively seek FASD cases, we won't find the majority of them. This also is true with some other common disabilities. But we're focusing on FASD here tonight. And in developed societies, the milder developmental disabilities, problem behaviors, might be mitigated. In other words, if you have a kid born with some pretty bad fetal alcohol damage in Italy, because they get adequate nutrition, they get stimulated in those hyper schools, and they get good education, and the parents nurture them well, they may well have been born with the potential of being partial or full-blown fetal alcohol syndrome. But through this kind of stimulation, brain plasticity, changes, improvements in their behavior, they wind up in the low normal range. And there's this one classic study that was done in Sweden, in 2 counties in Sweden, that legalized high alcohol content beer for 2 years. And they followed those kids born in those counties and the surrounding counties where they might have been crossing the line. Remember the old dry county days? Crossing the line to get the beer and taking it back. So they looked at the kids born in those 2 counties and excluded the kids born at that time in the surrounding counties, compared them with all the other counties in Sweden. And guess what? They were all less accomplished 30 years later. They had lower education levels. Their jobs were far inferior to the accomplishments of the other ones. Indicator after indicator, 30 and 40 years later, the kids weren't doing as well. But they weren't diagnosed as full-blown FAS. So they had made some progress, but not enough. So finally, what's the significance of all this? There were people 30 years ago saying that prenatal alcohol exposure is the leading cause of mental deficiency in many societies throughout the world. I didn't used to believe it, but in the last 15 years, I really fully do believe it. I think it is the most common form of mental deficiency in the United States. In 1981, the Surgeon General, Everett Koop at the time, made a very, very strict warning about fetal alcohol syndrome, and we all thought he was pretty crazy. Surgeon General advises women who are pregnant or considering pregnancy not to drink alcoholic beverages and to be aware of the alcoholic content of foods and drugs. That was reviewed just 5 years ago to see if it was still true, or are we smarter than that now? All of the evidence was put together by NIH, and they said, it's still true. There is no threshold. There is no safe one because of this individual variation. There's some women who can drink some, get away with it, fine. But there are other women who can't get away with it at all. So that's the safest advice. We have a newly funded study, which Beverly has advertised quite a bit, which has come to NRI to us. And we're studying fetal alcohol spectrum disorders in 3 states of the Union, Montana, South Dakota, and North Carolina, in 1 county or 1 major town in each one. We may be able to expand it out to some other jurisdictions too, depending on how we manage our money. But what we're doing is looking at fetal alcohol spectrum disorders as we did in those pilot studies in the northern plains in the West. And we're actually going to rule out a number of known genetic anomalies. So those guys in Sweden who criticize us as finding only genetic anomalies and calling them FAS, we can either eat our words or make them eat their words. And so we're looking for an improved understanding of maternal risk and protective factors in the general population of the United States. We have them pretty well nailed for South Africa, but we don't. So finally, I want to thank my wife, who's sitting right here, for putting up with all of my travels over the years and all of this. She for many years put up with this, and she still continues to put up with it, and I want to thank her. And so basically, my quest will continue for a number of years here until my body wears out and I can't do it anymore. But so that's thank you for listening, and if you have any questions, I would be very happy to try to answer them if I can. Thank you. Yes, sir.
One of those groups that had flyers said green alcohol. I heard green tea was good for you, but I heard green alcohol.
One of the flyers said green alcohol?
The flyer said green alcohol.
Really? Oh, it said geen alcohol. That means no. That's an Afrikaans word for no, forbidden. There you go. Yeah, G-E-E-N. It's Afrikaans word for no. It's forbidden. Forbid you to drink alcohol. Good point, though. Afrikaans words are tricky. I don't speak enough Afrikaans to much get by, but I do know enough to stay out of trouble. Very little, very little. We do find in our school studies, though, and we were very much surprised, particularly in that one set of pilot studies we did up in, I'll tell you the state, it's in Montana, but the kids were they really enjoyed getting the exams. They would take home and they would tell their parents, and it would feed back to the school board, and it fed back to the parents, and we got all kinds of accolades just by paying attention to the kids, screening them, and finding that most of them were normal. It just really was a good way to disseminate the information on a family-by-family basis. But no, it's not getting out much. I think in the United States and Europe, we think, well, we're too smart and too healthy and too good to have this problem, but I'm afraid the problem is there.
In your study, you're saying you're doing 3 states, North Carolina being one of them.
It is.
How are you going to go about doing that? How do you project to reach or find that population?
We're doing one whole county. Out of the 100 counties, we're doing one whole county.
Which one?
Yeah.
I think we'd better keep that quiet for now. I did say another thing, too, which is important. If another city or county in North Carolina wants to participate, I would be willing to talk to them. But communities are very sensitive about this, and they're happy to participate, but they don't want to be plastered all over the TV. because people think, well, gee, they picked it because they have a problem. The way we pick these cities and counties is, remember, I'm a demographer. So I go to the county profiles to find ones that pretty well mirror the socioeconomic status, the economic status, the educational status, those sorts of things of the general population of the United States, and the racial admixture. And the county we're doing in North Carolina is a little bit shy on some of the racial admixture, but income and everything else is pretty comparable.
Will we know later what state, what county was?
I'm sure. I'm meeting with the school board of that county to get the final go-ahead actually next Monday night.
So we can follow you around and see where you go?
If you're bored, yes. But, you know, I've got to talk to the school board first and respect whatever wishes they have. If they want me to keep it completely on the QT, I will. If they want me to advertise it, I will. It just really, it's up to them. I'm a guest in their backyard.
Phil,
Yes.
I'm assuming that with well, you're going to have parental consent, right?
Yeah. All these kids have parental consent. We will not have a kid participate in the study unless they have a signed parental or guardian consent. But we've been getting, most people say, well, how are you going to get that? No community in the United States is going to do it. We've gotten 70% in this town in the West, yeah.
And so the parents, it's not hidden, you know, is the...
Not within that county.
alcohol part of your research hidden within the larger nutrition health study, or is it real clear that it's focused on fetal alcohol syndrome?
We have to be clear that that is one of the things we're looking at. But we are looking for other things. A number of the tests that we do discriminate between kids with ADHD and normal. They discriminate between kids with genetic anomalies, some of which are very hard to diagnose if they're mild, like Williams syndrome. So we're looking for all of those things. That's why my dysmorphologists are human geneticists. They're broadly trained in all the teratogenic effects, but also the genetic effects.
And I was just wondering if a parent who knew she had consumed a good bit would be less likely to give her permission, but
Yeah.
then there are other advantages to having the testing done.
There are, and that's exactly why we try to oversample the small kids, because the vast majority of the kids are going to be in the small population. So if we only get a 60% approval in the small kids, we're going to get a higher proportion of those than we would in say, the larger kids. And that's one thing. The other thing is, every child that we see, we have a relationship with the parents. When they consent to us, we give them back the results of the battery of testing that we do, which is usually 2.5 to 3 hours. And 95% of those that we give back, your kid's doing fine. Be proud of that kid. And that really is great. That's the payoff to the parents. We also give them like $20 Walmart cards, things like this. And, you know, that's strange to me. 20 years ago, when you went to a human subjects ethic committee, you can't give them $20 to participate in study. You're buying research. You're buying data. Now, if I go and don't give a $20 this or $20 that, you've got to reward them for the time, the burden. So we have to go with all these approvals. We have to meet HIPAA compliance. We have to meet all these other things. But what we have to do, and my team is great at this, they really like kids. They really like working in the communities. We have to go and gain, we have to establish a good rapport and a good relationship with that community. We gain their trust that way. And it's therapeutic for some women who did drink, because, wow, Johnny's doing fine in X, Y, and Z areas. There's this one area that he might need some help, and we give that report back both in writing and, if we're concerned, orally, either through the school psychologist or through my own team, who are experts in this. And so we tell them what they can work on. If they care to, they can then release the report to the school. But if they don't want to, we don't release it. They have to sign off to release it. So people are a lot more altruistic and cooperative and positive, enthusiastic than we sometimes give them credit for. And I've really been kind of blown away. about that. This community we're working with in the early pilot studies, they asked me to do it. Because we were doing referral clinics up there because we were afraid to ask to go in the schools. But this one public health nurse 5 years ago said, well, come give a lecture to our physicians and this. And I described our in-school studies. They said, well, gosh, if you're doing that in South Africa and you're doing that in Italy, why can't we have that? And it's worked like a charm. So I have to admit that I was surprised by that. I really enjoy these these communities. I get beaten up periodically by some people, but you know, it's worked out well.
Question from online. The person says, I noticed you mentioned clinical interventions. What is being done, if anything, in private practice doctors in terms of prevention?
Boy, it varies. It varies for both obstetricians and pediatricians. Most pediatricians are very aware of it and are very careful to recommend many positive things about child development. Most obstetricians are too, but there are some mavericks out there, obstetricians who are saying that, well, maybe a little bit won't hurt you. And they go on, it seems like every year on the news there's one of these that occurs where an obstetrician will be interviewed by Dr. Oz or somebody, and they'll say 1 or 2 drinks is okay, and then there's this flurry of attention. But by and large, I think the information is getting out there. But sometimes it's too late because the women who are most likely to drink during pregnancy are the least likely to come in early for prenatal care. So it's happening. I think we have to do, in most areas of prevention in public health, it's pretty well established that if you want to maintain a positive behavior, like not drinking and driving, you have to do an inoculation and a re-inoculation every 14 to 16 months. It's pretty well established in a lot of areas. So we have to do that. There's a National Organization on Fetal Alcohol Syndrome. There are March of Dimes with birth defects. All of them are very, very good at getting out this information. They have pamphlets available. They work with healthcare providers of all kinds. And the acceptance of that is, I think, all over the place. Some it's very well received. Some it's not as well received. But I think, by and large, it's going pretty well in the healthcare system. It's those people who don't go in for early prenatal care and don't take the best precautions is the ones that we're not reaching. And what we've seen in South Africa is if we don't reach those highest risk minority of the population, it may be only 2 to 4% of the population we have to reach. But we're probably reaching the other 95% pretty well.
How did you define drink? I'm sure there's a standard for it, but how did you define what a drink is?
Yeah, the American standard, and it's a little different than the European and worldwide standard, is a ½ ounce of absolute alcohol. So a 12-ounce can of beer at 4.5 to 5% alcohol is a standard drink. A shot of 86 or 80 proof whiskey, 1.5 ounces, is a standard drink in a mixed drink. A 4 to 6 ounce glass of wine is a standard drink on the American system. In Europe, they do it on grams of alcohol, and it's actually a little less. Yes?
This is for you. In all those countries, all the food is different. Don't you have to get adjusted to all that? It ain't hard.
Well, let's put it this way. In the last decade, I've gained about 20 pounds, so I must have adjusted pretty well. Yeah, it's not bad. We're in these countries 2 to 3 weeks at a time. It's not like we live there over periods of time. I have to say that the question I'm asked most about that is people in South Africa always say, do you like coming to South Africa? And I say, no, I actually hate coming to South Africa. I like being here, but the trip's a killer. The trip is an absolute killer. It's 30 to 32 hours in transit, 22 hours of that on an airplane, and it's awful. But I do it 3 times a year. That's why I had to thank my wife. I mean, she puts up with that. Sure.
And maybe it was just me, when you showed pictures of kids, it looked like the left eye was more droopy. Was that accurate or just later?
No, ptosis, one eye that's more droopy than the other, is a very common feature. That's one of the minor anomalies that our physician...
The left eye?
No.
Oh, okay.
It could be either side. If it's both eyes, it's a condition called blepharophimosis, where the actual muscles of the eye are weak. And I didn't put it in here, but I've got some kids where they can hardly see. They're like that. Some of the really severely damaged ones. But ptosis, and also you notice in a lot of the kids, they had one eye that was going in the wrong direction, strabismus. That's more common because that indicates that the brain damage has not been bilateral. That one side of the brain is more damaged than the other. Yeah.
This sort of personal question. You said thin lips indicate lack of brain development. Does that apply in other places?
Well, I think there are two parts to that question. There are a lot of people out there with thin lips.
That's why I said it's personal.
There are a lot of people out there with thin lips that have completely normal development. That's why we have three parts to the diagnosis, and the third part is to find out what kind of maternal risks went on. A couple of the pictures of my clinical team and my sponsors that I showed you, some of the people have extremely thin lips, and we always kid them about that. But they're doing fine. It's a probability kind of thing. You have to put in all of these small features together, and if you get a high dysmorphology score, then there's a lot of reason to suspect. But if you only have one or two of these things, it's not an issue.
So we have learned that you really should be taking a stipend from those companies that sell the lip plumpers, right? That's what we need to do.
Well,
We have time for one more question if somebody else has a question.
The gynecologist advise women not to drink during pregnancy.
yeah, vast majority do.
And other kinds of addictive drugs, do anything like that?
They do. Do you remember about 20 years ago, there was this big publicity campaign and a lot of scare that was going on about crack cocaine babies? The fellow who started that and recognized that crack cocaine babies had a lot of anomalies like this and poorly developed, actually, when he published his first couple studies, he was actually diagnosing fetal alcohol syndrome kids who had actually been exposed to cocaine as well. And there are two different types of damage. Cocaine is a vasoconstrictor and it actually cuts off the blood supply to developing organs, like particular parts of the brain. So you have little voids in the brain. And if a mother is exposing her kid to both cocaine and alcohol, then you get this effect that he was calling crack cocaine babies. He's since backtracked and he's now an expert on fetal alcohol syndrome with some cocaine exposure. But actually, it's hard to understand. It's hard to accept, I should say. But alcohol is worse than marijuana. It's worse than heroin. It's worse than cocaine for unborn babies. It's because in large quantities, and it's legal and readily available, it's more teratogenic. It causes more damage. But the worst case scenario is when they use both. In the United States, we see that. That is that there are more multiple drug exposures in the United States than South Africa. That's why it's been so insightful for us to work in South Africa, because it's really only alcohol that's doing the damage.
What about breastfeeding?
Breastfeeding is another controversial area. A very high percentage of the alcohol that the mother takes in and has in her bloodstream will pass through the milk to the baby, and the baby's brain is growing like crazy the first 2 years. So you don't want to do it. You don't want to have a person breastfeeding and drinking at the same time. But that's another one that obstetricians for many years encourage some light drinking, particularly beer. And they still do that a lot in Europe, especially in the United Kingdom. But most of the experts, particularly in the United States, say don't do it because the alcohol goes through. And the brain really of a baby is developing very, very rapidly in the first 2 years.